Repeat the question. Thanks.
I am audible, sir.
Yeah. You are audible. You're audible.
Sir, is it possible to identify particular gene sequence via the in situ fluorescence hybridization?
Yeah. So thanks for the question. Very interesting one. Yes, mainly because we can design the sequence that the probe map accordingly to the laboratory request. So to give you an idea, for example, for HER2 gene, the probe is not mapping the entire gene, but a portion of 200,000 bases. Obviously, it is possible to identify specific sequences, but not very short one. There are some probes called oligoprobes that are now, let's say, under development from other companies, not us, or RNA-based oligoprobes that are shorter, but let's say the variability and the reproducibility of results is affected by the short dimension of probes because they are not so bright in evaluating the result. But for sure, the sequence is possible to design specifically a probe to detect a specific sequence. Also, if it is not, let's say, commercial available, it is possible to do so.
Okay, thank you, sir.
Thank you.
Sir, what type of microscopes are used to study FISH for chromosomal aberration study?
Okay, so regarding the microscope, obviously it is needed a fluorescent microscope that has it's the kind of apparatus like the fluorescent lamp and some filters and so on. So, but basically easily Googling, for example, Olympus BX51 fluorescent microscope, you can find all what you need. It is a so the let's say the cons of the FISH is that the microscope that you need is quite expensive. It's not a normal microscope, but the results are way better than with the silver in situ hybridization that use a standard microscope. So that the needs.
Okay, sir. Thank you. One more question. How this FISH is used to distinguish between elastogenic and angiogenic micronuclei?
Okay, so it is basically all about how we structure the probes and in particular the fluorescent molecules that we link to that probes. For let's say for more cytogenetic studies like bending or similar, there is a specific chromogenic technique that it is called painting, chromosome painting, that use an elaboration and let's say there is a need of a different microscope type with a different software that elaborate that image that use at least 5 fluorochromes linked to the probes. So with FISH, with the standard, let's say, FISH techniques, you can evaluate easily rearrangements, easily fusions. Double minute chromosomes can be accounted because of the pattern that they display, insertion, and so on. Big other type of alteration that can be traditionally evaluated with bending requires this chromosome painting technique that, to be honest, is not so common in laboratories. So there are few laboratories that are doing that, probably because it's way more expensive and way more difficult to be done.
Okay, sir. Thank you so much.
So,
Hello.
yes. So, what are the techniques to optimize Hello?
Hello? Yeah, I lost you.
Yeah, yeah. So I was asking that what are the techniques to optimize the process?
Okay, so other techniques to optimize the process, basically, to optimize, if you mean to optimize fluorescent in situ hybridization testing, we can act on the pre-analytical conditions. So once the samples are biopsied or surgical removal from patients, to reduce the so-called ischemic time. So just put those samples, ask to surgeons to put the samples as quick as possible in the formalin box. Maybe if they are big sample, just to cut them in half to allow formalin to penetrate. The other step is the fixation in formalin. You need formalin, obviously 4% formalin, which is the standard for histological procedures. But to ensure the correct time lapse for the fixation, so not less than 12 hours for small biopsies, not more than 72 hours, because otherwise the sample cannot be processed. And it is probably it is worse to have a very hyper fixed sample than a very low fixed sample than a standard one. So if I have to choose, it's better to not over fix it, the sample. The second procedure is to try to standardize the if you have kits, commercial available kits, is fine because they are manufactured under ISO certification, standardized procedures, and so on. But for 20 years, I made pre-treatment kits by myself in our labs. It is just to use the normal standard, let's say, good laboratory practices and try to standardize as much as possible the procedures. So try to avoid 5 different protocols for 5 different types of cancer. So just try to have one procedures with one set of reagents, and then work on that, changing probably the treatment timing, the concentration of the enzyme, and so on. So don't work too many variables because it is crazy. And then try to do to avoid the frequent freezing and defreezing of probes, because this is what can kill probes capability to work in the right way. Commonly, the number of cycle of freezing and defreezing of the probes can be 10 times. So if that means that if you have to do one test only, is not probably wait for a batch of tests to reduce this number of freezing and defreezing cycles. The other optimization can be done in the microscope visualization. As mentioned, there are some semi-automatic systems, but right now they need always the operator, a trained operator, because the operator needs to check if those systems work well. If I can say, there is no better system than human power or mankind, let's call it. A trained operator can be way more efficient than an automatic system, according to my experience and my colleagues' experience. So if you want to optimize the system, just select 1 or 2 operators that you want to train them for 3 to 4 months, daily watching and in situ hybridization slides at the microscope. After that, they are very well trained, and so you have optimized the procedure. So this is important, not because of the pretreatment impact the quality of the slide that the operator will go to read, but a trained operator can assess a slide in something like 10 minutes, respect to a not trained operator that can take 1 hour or more to assess the same slide. And we can support, obviously, if you have in your laboratories any specific need, just contact me. It's something that we do, and we are happy to do, to try to optimize and provide suggestions and advices to laboratories that need to set up also fish procedures or want to optimize fish procedures.
Hello.
Hello?
Sir, is there another biomarkers that can be studied under fluorescent in situ hybridization for breast cancer, apart from HER2?
Yeah, there are some others. There are TOP2A, which is another marker that has been used in the past mainly to evaluate if a polysemy, so a simple gain in the HER2 copy number, was a gain or an amplification. And basically, so from a treatment point of view, so consider, let's say, sorry, I just make a step back. Fluorescent in situ hybridization for HER2 for breast cancer started from a clinical need. So they say that there are this amplification, and they based on the therapeutic need, therapeutic target, the elaborate, let's say, HER2 as a key markers for breast cancer. Because of the key treatment in the HER2 in the breast cancer is based on HER2, it remained the main markers for breast. Obviously, let's say what we learned from other tumors type, HER2 is a way, but you can detect also EGFR, which is the epidermal growth factor receptor, can be positive in the breast cancer samples, at least because the HER2 protein is not create homodimers in the surface to activate the signaling, but always match with EGFR. So by evaluating by FISH EGFR amplification, you have additional information on the tumor. You can also evaluate eventually, but this is more for, let's say, if you want to start a research project, I would start about ALK or ROS rearrangements, taking let's say the point from reverting what is happening in lung cancer. So lung cancers that were or that are ALK positive for rearrangement of ALK or ROS1 positive for rearrangement, develop resistant pharmacoresistancy to specific treatment by amplifying HER2. So the treatment select HER2 amplification in the lung cancer sample. So if we would like to revert it, we can check, but this is a completely new project that I'm suggesting. We can check if a patient with HER2 resistant, positive resistant cancer may have developed some of ALK or ROS1 rearrangement. Another marker that can be useful, but in male breast cancer, not in female breast cancer, in male breast cancer, is the gain of the X chromosome and the androgen receptor. We did a study, I did a study during my PhD, and we demonstrate that males that have a gynecomasty with a gain of X chromosome, they had a higher risk in developing a breast cancer. And in the male breast cancer is a, is a low number of cases, obviously compared to female breast cancer, but because of several reasons. First of all, males are not doing breast cancer screening, at least in Europe. It is a type of cancer that is diagnosed as a very aggressive or late stage. And so we identified that patients with gynecomasty, if they undergone to fluorescent in situ hybridization for X chromosome centromeric specific probe, they have a gain. It's better to follow them up any 6 months to avoid the occurrence of aggressive breast cancer. So that are the markers that I feel can be useful in breast cancer together with the HER2 gene.
Yes, sir. Thank you, sir. Thank you very much.
You're welcome.
Hello, sir.
Yeah.
So, what kind of probes are best to use?
In breast cancer or in general?
In general.
Oh, obviously, it's really depends on the tumor that you are going to analyze. So, for example, as mentioned, right now guidelines for breast cancer includes HER2. For lung cancer, they are NTRK1 and TRK2 and TRK3, ALK and EROS1. For brain tumors, there are new guidelines that amplify the number of FISH probes and markers that you need to test for a single patient, starting from the codeletion of chromosome 1p and 19q, CDKN2A and 2b, the deletion of CDK, the EGFR centromer 7 gain, and the loss of 10 and the centromer of the chromosome 10, and the BRAF rearrangement probe. They are for diagnostic usage. I'm not touching the leukemia and the blood tumor alterations because they are the diagnosis of that and also the evaluation of recurrences are based on FISH probes like BCR-ABL, EGH, rearrangements, and so on. So it really depends on if it is an R&D project, on the tumor that you are going to analyze, if you're starting from that tumor. If it is for diagnostic purposes, it is better just to check the guidelines and have it down, take it down from that guidelines, the markers that you need. Again, I would like to say, if you have any specific request, just find out, try to reach me out. I try to respond as much specifically as I can, so we can address any specific needs.
Yes, sir, sure.
Thanks.
Sir,
Yeah.
how to study gene alteration using fluorescence in situ hybridization?
I didn't hear you very well at the beginning of the question, so if you can repeat, sorry.
Sir, how to study gene alteration using fluorescence in situ hybridization?
It's a nice question. So I never think about it, but I will have to think about your question. Sorry for not able to providing a quick response to that, but so please messaging me, email me about that question. I will try to help you and to providing an adequate response for that. So sorry for that.
Okay, sir. Thank you, sir.
Sir?
Yeah.
Yeah. So, what are the applications of FISH beyond the cancer study?
Okay. So we find application also in veterinary pathology. So we spoke about breast cancer in humans, let's say. We find application in veterinary pathology, for example, not only the cats and dogs breast and lung cancer, but also in the microbiome study for certain type of bacteria that can be retrieved in the gut of certain dogs breeds. Can be studied with fluorescent in situ hybridization. Obviously, microbiology can be, let's say, investigated with fluorescent in situ hybridization. In particular, if you have to localize the specific cell in which pathogens are. For example, the Epstein-Barr virus is it is used in situ hybridization to identify the presence of the genome of the Epstein-Barr virus in a certain type of cells that are relevant for cancer and for the cancer treatment in that case. But for HPV, you can do that, and so on. Someone did something for during COVID pandemic to localize COVID, let's say, RNA fragments in the lung specimen from after autopsy to try to see where specifically COVID virus migrate in the lung in patients' lungs. So there are, wherever you need to, let's say, specifically identify DNA or RNA strand, and you need to localize specifically cell by cell the presence of this strand, you can use fluorescent in situ hybridization. So also vegetables, the NGO, the GMOs, vegetables, if you need to identify the presence of a gene in certain varieties, you can do that, and so on. So wide range of application. Happy to let's say to check new application for fluorescent in situ hybridization also.
Yeah, thank you, sir.
Hello, sir.
Yeah.
Sir, which type of biomarkers are very important in the breast cancers?
So as mentioned, HER2 is the crucial one. Then you have EGFR, so epidermal growth factor receptor. From immunohistochemistry point of view, you need to test the estrogen receptor, the progesterone receptor positivity. Some centers start testing BCL2 and P53, but they are not so it's a second level. If you want to do more, it's possible. And for certain type of breast cancer, need to test also androgen receptor positivity by immunohistochemistry, obviously. Estrogen receptor, progesterone receptor, and androgen receptors by immunohistochemistry. HER2 immunohistochemistry, and then FISH, and EGFR also in immunohistochemistry or in FISH.
Okay, sir. Thank you so much. Thank you so much.
You're welcome.
Thank you, sir, for words of wisdom and making us familiar to the advanced technologies which will enhance and open up new opportunities in research field and pave the path of success.
Thank you.
Our next speaker is a profound researcher, Dr. Deepak Raut, who will shed light on nanotechnology-based development of medicines from herbal drugs. I now request Rutuja More ma'am to kindly introduce our speaker. Over to you, ma'am.
Thank you, Shraddha. Am I audible?
Yes, ma'am.
Okay. Good afternoon, one and all. I, Rutuja Mure, Assistant Professor, Sihagarh College of Pharmacy, Vadgaon, Pune, welcomes you all for this session. I am profoundly delighted to take an opportunity to introduce our today's guest speaker, Dr. Deepak Raut, sir. Sir is currently working as associate professor at Amrutvayini College of Pharmacy, Sangamner, in Department of Pharmacognosy. Sir has completed his PhD in 2015 from Jadavpur University, Kolkata, and his M.Pharm in 2006 from Savitribai Phule, Pune University, Pune. He is recognized as PhD supervisor and PG teacher of Savitribai Phule, Pune University, Pune. He has taken grant of BCUD for 4 minor projects and grants of BCUD for innovation programs. Sir also has one published patent on his credit. There are 12 international and national publications in his credit. He is a life member of Society for Ethnopharmacology India, Society of Pharmacognosy, and Research Scholar Hub. With this brief introduction, I would like to invite our guest speaker to start with this session. Over to you, sir.
Hello, Raut sir is just now he putting in the session. So, Shraddha, can you please again introduce?
Yes, sir.
Raut sir just now joined the session.
Yeah.
Yeah. So, can you just start it again? The session?
Yeah. Yes, sir. Our next speaker is a profound researcher, Dr. Deepak Raut, who will shed light on nanotechnology-based development of medicines from herbal drugs. I request Rutuja Mule ma'am to kindly introduce our speaker. Over to you, ma'am.
Thank you, Shraddha. Good afternoon, one and all. I, Rutuja Mule, Assistant Professor, Siaghad College of Pharmacy, Vadgaon, Pune, welcomes you all for this session. I am profoundly delighted to take an opportunity to introduce our today's guest speaker, Dr. Deepak Raut, sir. Sir is currently working as Associate Professor at Amrutvahini College of Pharmacy, Sangamner, in Department of Pharmacognosy. Sir has completed his PhD in 2015 from Jadavpur University, Kolkata, and his M.Pharm in 2006 from Savitribai Phule, Pune University, Pune. Sir is recognized as PhD supervisor and PG teacher of SPPU, Pune. He has taken grant of BCUD for 4 minor research projects and grants of BCUD for innovation programs. Sir has one published patent on his credit. There are international and national 12 publications in his credit. Sir is a life member of Society for Ethnopharmacology India, Society of Pharmacognosy, and Research Scholar Hub. With this brief introduction, I would like to invite our guest speaker, Dr. Raut, sir, to start with the session. Over to you, sir.
Yeah. Good afternoon, all. Good afternoon, ma'am. Am I audible?
Yes, sir.
Hello?
Yes.
Yeah. Let me share my screen. May I know, is it visible? Yeah.
Yes, sir.
Yeah. Shall I start then?
Yes, sir. Yes.
Yes. Thank you, sir. Thank you very much. Good afternoon, all present over here to listen today's conference sessions. I, Dr. Deepak Raut, just joining you right now to introduce a small topic in our pharmacy, that is nanotechnology-based development of medicines from herbal drugs. So, inside of these herbal drugs, just I would like to add some basic information from where we have started these herbal drugs. That from the quote from Hippocrates, that is, our food should be our medicine, and our medicine should be our food. So we have a lot of basic sources of medicines from plant sources, and it is from traditional point of view, and there are different traditional categories from where we have started our medicines developed from the plant. The main source that we all know, that Indian traditional system of medicine, that is Ayurveda. Then traditional Chinese system of medicines are there, Arabic Unani medicines are there, homeopathic medicines are there from Germany. And these peoples have introduced natural source. We can synonymously call as a natural source. Here you see there is a development of natural products, their medicines from plant sources. Right now we know the different medicines that are obtained from plants, but they have very good market or very potential uses in medicine, like morphine, caffeine, and other medicines from like polyphenolics from different fruits. And we have a lot of evidences of herbal medicines, that is from integrative medicines. Nowadays, there are a lot of informations available under the complementary and alternative medicines, where people have used plants as well as other natural sources for the medicines. The development of medicines was due to some evidences, that is discovery of penicillin, isolation of resistance, basically this from plant, anti-cancer properties of vinca plant is there, discovery of dioscorides, that is to prepare semi-synthetic steroidal hormones. So along with this contribution, just in recent knowledge of the medicines from herbal drugs that we call as the modern medicines, we call the chemicals which are potential for its uses, that maybe that could change the physiological action, or that could have the pharmacological effect on human beings alternatively. And we have started its research from isolating the compounds from the plants to cope up with the modern medicines. So in this case, that we know, we have that natural product chemistry to search of new lead compounds from the plants, which there are involvement of isolation, structural elucidation, molecular degradation, and the biological activity of that particular compounds. So this is a representative example that having anti-infective as well as anti-cancer compounds from the plant sources. And for this purpose, we are using their different isolation technique to get one particular compound which are responsible for that particular disease or particular physiological action. So here, in case of complementary and alternative medicines, which is a group of diverse medical and healthcare system practices and products that are not presently considered part of conventional medicines. The complementary medicine is used together with the conventional medicines actually, and alternative medicines is used in place of that conventional medicines. So purpose of showing the slides, initial slides, are that there is the importance of the herbal drugs in medicine, and there are a lot of limitations we are just facing in the world. Even though WHO have also faced some standardization issues of herbal medicines, but still the herbal medicines are very potent. In India, we use these herbal medicines as drug. And in the different countries, we call as a nutritional supplements. So these herbal drugs, which are known as nutritional supplements instead of these medicines, mostly in developed countries, that are nutraceuticals or the nutritional supplements. The term nutraceuticals was developed, coined by Stephen DeFelice, which called as a combination of word of nutrition as well as pharmaceutical. That is a pharmaceutical form or dosage form of the nutritional supplement which are present in plant. It covers the isolated nutrients, dietary supplements, and we having the issues like issues of the herbal formulations, like the relatively low as or variable bioavailability as a result of various physicochemical and or physiological processes, which have the potential benefits of many herbal pharmaceuticals, which are not optimally realized. So there is need to modify, need to transform such type of herbal drugs that we call them as a supplements or herbal extract or powders, whatever that we used as per our traditional knowledge, Ayurveda or other traditional knowledge of the plants. The main focus is here, it's relatively low and or variable bioavailability that are especially due to the some reasons. Basically, the reasons are there is formation of insoluble complex with the components in the gastrointestinal tract. then low solubility in gastrointestinal fluid, then low permeability across the mucus layer or epithelium cells, and molecular transformation in GIT, as well as restricted liberation from the plant materials. Yeah, that is the reduced BioD, or just due to that some issues of solubility and stability. Actually, we are expecting to have the viability of the components of the drugs or medicines. That may be single drug medicines or multi-drug medicines, or that could be our herbal drugs. Herbal drugs basically have a number of chemicals inside it. Chemicals inside it. So the reduced viability and stability, as well as the permeability of the bioactive compounds in the gastrointestinal fluid, leads to their partial absorption from GI tract during the first pass metabolism. This results in their reduced or almost no biological activity, which is major concern of the researchers nowadays. So this is the thing that in case of this conventional mode of delivery system, some part, the portion of the extruded drug either extruded or instigate non-specific toxicity and adverse effect owing to its understandable biodistribution, and portion or some part of the drug from that conventional mode of delivery that are administered doses absorb and reach the actual pharmacological site of action. So here we need to find the solutions for the portions which are get excreted immediately, or which are not reached at the site of actions, or which are producing the toxicity as well as adverse effect. So this is there is need for development of new drug delivery technique, which could have that effective solubilization, encapsulation, and delivery of that actual bioactive compound that leads to excellent bioabsorption in the lower dose, which could produce less toxicity or no toxicity. Then reduced frequency of administration, as well as a maintained therapeutic index. So this is BCS classification that we normally use for most of the allopathic or synthetic drugs that we have today in modern medicines. But in case of herbal drugs, actually we couldn't follow this type of system, BCS classification system for the herbal drugs due to its failure. Actually, this biopharmaceutical classification scheme that is widely used in pharmaceuticals to characterize the major factor limiting the oral bioavailability of the drug, that such as their low solubility as well as low permeability. And there is various complicating factors that impact the bioavailability of herbal drugs present within the plant matrices present in the extract, which limit the usefulness of the BCS classification system for most of the herbal drugs as well as some of the food components. So for this purpose, which are the factors that are actually limiting the bioavailability? There are three major classes have been framed for this purpose, that is bioaccessibility, absorption, and transformation. So in case of bioaccessibility, we have that liberation limitations, solubility limitations, and interaction limited the transfer of the drug. Then in case of absorption, that is a mucin layer transport limited absorption. Then there is a tight junction present in our cells GI tract or the other cell surfaces. Then membrane permeability limited, even though the drug is there in blood, it could have the problems with membrane permeability. As well as active transport and influx transport also plays a role over here. In case of biotransformation, actually the drug, the chemicals or components we required, which has some biological activity, that could get degraded. That is, the degradation due to chemical degradation is there, and as well as due to regular fate of that particular drug in the body due to in liver or in other part of the body. That is metabolism limitations are there. These are the pictorial description of the liberation from the food matrices, then solubilization in intestinal fluids are there, and then interaction and insoluble complex formations are there. So this bioaccessibility, which is related to the herbal pharmaceuticals within the intestinal fluids. Before ingested, that particular drug can be efficiently taken up by the body, and they must be present within the physical form that is suitable for absorption. In case of hydrophilic herbal drugs, this may simply the fractions of the drug that is fully dissolved within the intestinal fluid rather than present in an insoluble form or complex form or crystalline form. In case of hydrophobic nutraceuticals or other hydrophobic drugs, this may be the fraction that is solubilized within the mixed micelle in the small intestinal fluid. This is a liberation limited factors. That is, bioaccessibility may be limited by its ability to release the content from the matrix, from the plant cell. For example, is the carotenoids, which are trapped within the cellular structure of the vegetable. That is not fully released from GIT after vegetable is ingested. Increasing the release of this kind of chemicals from the plant matrices would be an effective strategy to increase bioavailability. This could achieve due to that altering the plant processing conditions like homogenization, sharing. Even we are eating that. If we chew it properly, then we could get rid of this type of problem. Or just by altering the food matrix properties such as composition and structure of that matrices. In case of solubility issues, this is ability to be solubilized within the site of absorption that we call, that is in intestinal fluid. The hydrophobic drugs have low solubility in water, and therefore that need to be incorporated into mixed micelle in a small intestine before they can be absorbed. And bioavailability can be therefore improved by utilizing composition or structure that enhance their intestinal solubility. Then, in case of interaction limited, there is an interaction with the other components in GIT that could arise from ingested matrix itself, or that could be from naturally, which could naturally present within the GIT. That drugs could form or may form the insoluble complexes that is not easily absorbed, like anionic long chain saturated fatty acids that interact with the cationic calcium ion in the intestinal fluid and form an insoluble complex that has low bioaccessibility. These can be limited by presence of particular components within ingested food matrices, such as ionic biopolymers or chelating agents, like chelating agents that may decrease the bioaccessibility of the minerals, or the cationic biopolymer, like we have the chitosan biopolymers, may decrease the bioavailability of anionic fatty acids due to the formation of insoluble electrostatic complexes. In case of absorption, most of the drugs, like here allopathic drugs, herbal drugs that from the GI fluid, the fraction that travel through the mucous membrane across the epithelial cell and into the systemic circulation that causes this limitation factor. That is, mucin layer transport limited is there, bilayer is there, tight junctions are there, active transports are there, and influx transporters are there, which are just shown in pictorial form here in this slide. So we have this epithelial cell and mucous layer transports are there, then passive transports are there, which as well as tight junctions are there, which inhibit the travel of the desirable chemicals from the food as well as from the plant drug. This is another form, that is a transformation takes place in our body of that particular chemicals, and their activity get diminished. This bioavailability of many drugs is limited because of the transportation that are converted into inactive form. And this molecular transformation can be divided into chemical degradation as well as metabolism. Example, we can take that polyunsaturated lipids, fatty acids and lipids, such as omega-3 fatty acids or carotenoids. Actually, these compounds are very active in vitro, but they produces their limitations when we are taken it into our body, or it is injected into the body. That may oxidize due to presence of pro-oxidants in GIT, whereas proteins or peptides that may hydrolyze by highly acidic and enzyme active environment of the stomach. Then as I told, some drugs get metabolized by specific enzyme system present within the GIT or levers. So in case of GIT, this resveratrol, quercetin, and epicatechins are generally metabolized. Many important phytochemicals, which are they are susceptible to phase 1 and phase 2 metabolism within GIT. So these are limitations to get desirable biological activity or pharmacological action of the drugs, herbal drugs that I am focusing on. So we have to find out the solutions for that to get, because nowadays we have a lot of medicinal forms, pharmaceutical forms of the herbal drugs in the market, and unfortunately we cannot monitor their fate for all the drugs, fate of that drugs into the human body. They are not monitored because most of the herbal drugs given by the Ayurvedic practitioner, they are not going for a specific evaluation of the drugs, its effect, its side effects or effects inside the body. So there is myth that herbal drugs are there, so they are safe for body. Every herbal drug is safe. If herbal drug is there, no, anyone can take it, or that we call it does not produces any toxicity. But it is totally wrong for all type of herbal drugs. So in case of food materials, we can say like this. But from the food materials, we are interested in some phytochemicals. Phytochemicals shows its effect, its activity in a specific concentration that contain not desirable concentration of that active ingredients that we call as the medicines. So there is one important technology, that is nanotechnology, that we can use in medicine to improve the bioavailability of herbal drugs. Along with allopathic drugs are also tried with the nanotechnology, and we got more success in this case. So similarly, herbal drugs also can be transformed into a suitable dosage form by use of the nanotechnology, because when we use it, it extends the product shelf life, it enhances solubility, it influences the texture and flavor, that will enable sustained release formulations, then it reduces toxic effect. That could be the targeted smart delivery of herbal drugs, as well as that could protect the drug from the moisture, and ultimately that improves the bioavailability or the existence or concentration of the actual drug from the herbal drugs at the site of action. Actual chemicals which are responsible for activity are present in the herbal drugs, and that we can monitor if we use the nanotechnology. We can enhance that particular activity through the nanotechnology by this way. That is, herbal drugs could be loaded into some matrix or along with some excipients. So this is again the part where we can work. Most of the pharmaceutical peoples or those who are doing their basic research as well as PhD research or further research can work on that matrix or excipient that we are using, like most of the polymers nowadays we use in nanotechnology, which could be the smart polymers. We can we can modify that polymers as per our requirement, which will improve the aqueous solubility, stability, bioavailability, circulation time, as well as target specificity. So we have to solve the problems one by one, as that could start from the solubility, that could start that could go to this bioavailability, the circulation time. Later, the stability. Stability is a major issue in nanotechnology, actually. So the scientists are trying to solve these issues one by one. So why this nano drug delivery system for herbal drugs? So there are the limitations that already we have discussed associated with the current dosage form of the most of the antibiotics, most of the cancerous drugs, and for this nanotechnology have been developed. A lot of formulations are developed for the cancer as well as for infectious diseases. That inadequate drug concentration at target sites we are not achieving. This is a basic problem of cancer and infection. Actually, the drugs which we are using for these two, specifically anti-infective drugs or anti-cancer drugs, they produces toxicity for normal cell too. So we have to minimize their toxicity for normal cell, and we have to enhance their activity against that particular cell, like cancer cell or bacterial or viral cells. So that the adequate concentration should be there at target site, not at the other sites where there is no infection or there is no cancer. So if we could do it possibly, then we can decrease the toxicity of such type of chemicals. And the problems, therefore, it's actually the issues of that problem is delivery of the drugs, not the drug itself. Drug is very active. Actually, we have a lot of potent drugs. We have a lot of drugs, herbal drugs also available in market. But the delivery of the drug is not specific. So we couldn't get that type of action, or we couldn't reduce the toxicity of that drug. Okay? To kill that particular infection or to kill that cancerous cell, that drug should have particular toxicity to that particular cancerous or infective cells or bacteria or viruses, but should not produce any effect to the normal cell. So we have to find out the way to carry out this, and nanotechnology plays an important role over here. The drug efficacy is often transformed by non-specific cell and tissues biodistribution, and some drugs are rapidly metabolized or exuded from the body, and there is a need for drug nanocarriers that efficiently target that microbial site in the body or cancerous site in the body, specifically for these two type of disease. The nanoparticular based drug delivery system strategy is directing to advanced therapeutic efficacy of currently available herbal drugs that provides that outstanding achievement, and nano carriers, that materials, drug excipients that could be able to accumulate at the target site either through the passive targeting or active targeting on the drug location, dose, or recovery time. Due to this nano size, it can travel faster, even passively, or even that tissue permeability can get enhanced, or even we can target it at nucleolus where our DNA, RNAs, their synthesis are taking place, and protein synthesis are taking place, where it could interfere there and can solve the problems of cancerous treatment. So this is the nanotechnology where we could use for infectious disease, especially. That is due to that drug resistance, adverse effect, inadequate drug concentrations, poor patient compliance, and increased frequency of administration, that we can overcome this by the nanotechnology. So the nanotechnology overcome these problems, that improve the patient compliance, that could be targeted at delivery to infection site, that decrease side effects, and overcome this resistance in case of microbials. So these are the drug targeting. Another aspects of the nanotechnology, that is we could target the drug through the stimuli responsive properties of the drug. So there could be some chemical stimuli, present in body, and if we could target that particular stimuli, and we expect that interaction of the stimuli and our carrier, and carrier have our herbal drug or normal allopathic drugs, and if there is interaction, then there could be some changes in the structure of nanoparticles, nano formulations, and release the drug at the target site. So this similarly it could happens with the biological stimulus like proteins, enzymes, sugars, and it could happen some physical stimulus like light, temperature, and ultraviolet. A good example here is in case of infectious diseases. Actually, in case of these infectious diseases, what happens where the infections are there, if infections are localized, where these infections are there, it creates a different environment around that infection. Means first thing is that changes body pH, as well as some other enzymes of that particular bacteria or viruses are released at the site of action. If we possibly target this type of pH or that enzymes, then we can release our drugs from the matrix at the site of action only, where the pH at the normal body cells are normally it is normal at 7 or 7.2 like that, and at infection site it could reduces up to around 6.5. So we can choose such type of stimuli to target from our excipient or polymers or change polymers, and then we can release our drug at the target. This is one example that I gave you. These are different evidences of nanotechnology that already going on. Use of the herbal drugs in the formulation. The excellent example is the curcumin that demonstrate the advantage of nanotechnology for the incredible enhancement in its solubility and bioavailability. Actually, curcumin has it's a hydrophobic molecule. It has a very less solubility, and that's why it has some problems, issues to just transport or fit in our body or travel in our body. The potential herbal drugs with several significant properties that include anti-cancers that could enhance when we formulate them into the different forms like by using nanotechnology. The encapsulation of curcumin along with the magnetic nanoparticles exhibited excellent reduction in cancerous cell in vitro in the synergistic with that of the magnetic hyperthermia, which are already demonstrated, as well as lutein, which are also encapsulated, incorporated into polymeric nanoparticles, so as to enhance the solubility of that lutein. The cream using nanolutein and curcumin for cosmetic applications are also studied. So these are the examples how these nanotechnology-based formulations are effectively used, could be effectively used for this purpose. So here, in case of curcumin, just depending upon its different groups present in curcumin, like OCH3 or OH or C-O, we have to use that excipients or polymers for this purpose, and we can predict that accumulation or entrapment of that drug in the polymer if polymers are producing some self-assembly or 3D existence in the particular solutions. So when we study that one by one, how we can design that particular excipient for that particular drug, for example, curcumin or any sort of drug we can take. A lot of work has been carried out on the curcumin. So I have given this example. So another example is a carrot that which have carotene and have very hydrophobic molecule. Actually, we required, if we want to separate this carotene, beta carotene from carrot, we required highly nonpolar chemicals like petroleum ether or NHA, because due to that, as you know, that light dissolves, like the nonpolar chemical dissolves in nonpolar solvent. Means nonpolar means it is hydrophobic type of materials, and it is very problematic to absorb this drug from the body when we take this in any form. So we are not achieving our goal, that bioavailability of the drugs normally if you take it. So we can modify them into nanotechnology derived products, and then we can use it. So this is with the help of the emulsifier, the drugs can be incorporated into that by using the polymers, and which formation of the nanoparticles are there, then which create the water dispersible formulations, which having the high entrapment efficiency or efficacy of the drugs into the nano carrier, so that maximum amount or more amount of drug we can bring with that formulation or with that excipient and reach at the site of action. As well as that, that means that intrapid efficiency could be increased, and it process high reproducibility in this case. So in case of phytomedicines or plant-based drugs, we have a lot of examples of phenolic compounds. So these phenolic compounds having again limitations in their absorption. So we can increase, we can use this technology for such type of drug. For example, you take example of rutin and quercetin. Rutin has some sugar moieties over there, as well as quercetin does not have that particular sugar moiety. So the change in the fate, change in the absorption states takes place in this case, and we cannot control this little bit difference in rutin and quercetin, and we couldn't control their fate, their absorption of their pharmacokinetic in our body when we are given by the traditional route of administration. So the technique we normally have to use here is a chromatographic technique for plant-based drugs to carry out its research work using the nanotechnology. So this is the way why nanotechnology is used in herbal drugs. That is, this nanotechnology will develop herbal drugs and functional foods with distinctive trait would have the potential to deliver the unique product to the world. And the development of better characterized and research proven product will help to enhance the consumer confidence in natural product in the rest of the world. This was the reason that I have chosen this such type of research work. There are a lot of applications of nanotechnology in herbal drugs. So we know that there are different metal nanoparticles we could synthesize, we could produce. Then phytosomes are there in case of herbal drugs, liposomes are there, myosomes are there, other lipid nanoparticles like solid lipid nanoparticles, SLNs are there, NLCs are there. Then there are especially polymeric nanoparticles where we can use polymer in any one of the formulation type act as a carrier, and so on. So what happened with these formulations, nanotechnology based formulations, which increases the solubility as well as tissue permeability of the drug basically, and it has a targeted drug delivery. It could have the controlled drug delivery, as well as it might have the responsible drug delivery system. Some of the work that we have carried out here that I just want to focus, so that the researchers can take such type of formulation technology for the herbal drugs to enhance the acceptability of herbal drugs. There are a lot of limitations for herbal drug research out there, so we can just solve these limitations, or we can solve these research problems in coming days. So in case of these polymeric nanoformulations, we have prepared this pelargonium gravelone oil formulation. Actually, this is the essential oil, and generally it has some antibacterial properties. Because of its lipophilic nature, we couldn't monitor its absorption properties, and we cannot enhance its absorption properties. So basically, solubility issues are there. So we could use the polymeric materials. Actually, we have used the polymeric materials to modify them. Then we have done that some modifications in the polymer, and we have mixed it with oleic acid. Actually, there are some drugs, oils or fatty acids, which we can use as excipient, as well as we could use for basic enhancement of activity, because uric acid, these type of acids could have the similar activity in the higher concentrations, or low activity in that particular concentration they are using, that could support that actual activity of the drugs that we are going to incorporate in it. So in this increase the solubility that we got, as well as enhance the antibacterial activity in vitro at low dose we have found. The basic work was there, that we have taken that oleic acid. Generally, normally we can modify, we can attach this oleic acid to the one of the mostly used polymer, that is the polyethylene glycol. Actually, poly-PEG has different grades. So as from lower grade PEG to a higher molecular Dalton size PEG, like from 400, 700, 1000, 2000, 3000, 5000, like that. If you increase this hydrophilicity, its hydrophilicity decreases. And depending upon our drug which we want to incorporate, we can choose that particular polyethylene glycol grade. And here we have used that oleic acid as well as PEG to make its combination, so that it could have some self-assembly structure, and we could incorporate, we want to incorporate that drug into it. So there is a critical step that we have to choose that molar ratios of oleic acid or fatty acids, as well as PEG of different grades, so that that particular the chemicals could incorporate into it, as well as their you see their hydrophilicity and hydrophobicity, pH, that we call as the pH balance. Hydrophilicity balance should be there, so that it could form that complex. Sorry, it could form that self-assembly or nano size particle. And here we have just mixed it. We have made the reactions and formed this PEG oleic acid polymer complex, and that we have prepared the different formulations with the different grades and proportions, and we have finalized one formulation which could have some basic nanoparticle form tested with particle size analyzer. So this is the procedure. I'm not going in the procedure that we have adopted, and we have prepared this nano formulation without this polymer combinations, PEG 400 oleic acid, as well as we have taken PEG 3000 oleic acid, so PEG 600, 6000 oleic acid combinations, and we have prepared these formulations. As well as without these formulations, with only PEG, we have tried the formulation to prepare the nano formulations, and we got the results. Actually, this is very helpful tool for doing the chemical reaction, to monitor the chemical reaction. Simply with our TLC. TLC is one of the tools normal routinely is widely used tool for the monitoring the chemical reaction. Along with that, if we go for FTIR, then we can understand the reaction formation very clearly. Okay, here you know that in case here you have, you see in this reaction this oleic acid has large terminal COH group, and if you take FTIR of this,
sir, sorry to.
Yes. Hello.
Sir, slides are not changing. If I'm not, sir, slides are not moving.
Which slides is there?
What is that? It is nanotechnology based delivery that protects the against it. All parameters you have explained. Now we are at that slide only.
Okay.
But sir, am I right at your end?
No, actually, just I'm sharing it again. This one. Is it visible now? Does it
at my end, it is no. At my end, it is all parameters only. Improves the viability, then reduce the toxic effect only. Someone please help. Is there any issue at my end? I don't know then. But sir.
Hello, ma'am.
Yes.
Hello, ma'am. Ma'am, it's your network problem is there. I think
Yes,
slides
that's
are
why.
moving there.
Check it again.
That's why. That's why I mentioned whether it is at my end. I don't know. Okay. Thank you. Thank you. Sorry, sir. Sorry.
Is it
Sorry to
visible now?
bother.
Is it visible? Preparation and synthesis of
Yes, sir.
copolymer. Is it?
Yes, sir.
Okay. So you see, this is oleic acid. Actually, this structure of oleic acid have terminal COH. So we could easily predict whether the formation of the chemical reaction is there or not. And this sorry. Hello. Sorry. So the prediction of the chemical reaction over here, it's if you go and see that FTIR of folic acid, you will have the sharp peak for this ester linkage, and if you join it, then that terminal OH that peak get reduce or get change, and you can predict their reaction. And when you have reactant as well as product in your structure, in your solution, then we can easily separate it by using chromatography, column chromatography, or possibly preparative thin layer chromatography. I think most of the times we can use column chromatography for suitable drugs. And if those drugs are very difficult, then simple change in pH of the solutions we can recover that solution depending on the nature of it, properties of that reactants and product. So here we have used Udrigit and Queen as a surfactant and prepared the nanoparticles and say taken this particle size as well as with the PAG 6000 oleic acid polymers, we have checked it and we got this type of particle size difference, and we have checked it with a we have compared it with a plant that prepared without any oil or drug incorporated into it, and further we have evaluated it with SEM images. The surface morphology are carried out for this purpose. So here you see there are first figure that indicate that blank formulation which does not have any drug incorporated into it, and when we incorporate drug into it, then it forms at around 10,000 x. We got this type of structure of nanoparticles formations, as well as at a different site we have got a different type of particle, different resolutions of this, and we got that enhanced antibacterial activity of the drug in formulation form, in nano formulation. So in this project we have just prepared this modified, we have prepared the modified polymers, and we probably use it for the antimicrobial activity. So second sort of dosage form we can try, you can try for this. It is development of the dosage form for specific chemicals like polyphenolics. Already we have tried it for EGCG type of chemicals. Actually, we are not able to separate any one form of EGCG, but we have separated EGCG rich fractions which have different forms of the chemicals of catechins, and that we have tried to incorporate into the formulations. Because if you go to formulate any herbal drugs in their crude form or in their extract form or in the fraction form, there are a lot of limitations to monitor that drugs, as well as lot of chemicals are there. So try to separate that chemicals in reduced form, in reduced numbers, and if possibly if you could try for single chemicals, single potent chemical, then it's better than this. but it's not easily separable compounds from the plants. So we could use this strategy, that is that particular chemical rich fraction we can take, and we can prepare the formulations so that we can control that chemicals in the formulations as well as in the you can say in separated form from the formulations, like your dissolution studies are there. So here we have used these polyphenols because of its antioxidants, anti-cancer, antiviral, anti-obesity, and antibacterial activity. So we can choose any one activity over there, and we can work on that in vitro as well as in vivo. So this was our purpose, that is extraction of that EGCG, then polymer selections was there, then formulation studies and evolution of that, and in vitro activity of that formulation. So we have tried to separate these EGCG fractions. So we have check it, analyze it by thin layer chromatography, which we have separated from this column chromatography. Further fractionation, we have to again purify by using column chromatography. It's not a simple task, just only go for one column chromatography and you take it. No, there are a lot of chemicals can appear in one spot, that even you got that one spot on TLC, 3-4 chemicals could be there. So further you can purify that by changing the mobile phase of the column chromatography. So this is the formulation that we have prepared from that by using a copolymer as well as polymer and some modified polymers. So this is another type of formulations, nano formulations, that is nano emulsions, that we have tried with Withania somnifera extract. Actually, this was a little bit crude work that we have carried out, but we could achieve that getting the nano formulation, nano emulsions in very good form, and we have incorporated this Withania somnifera. One of the limitations for the herbal drugs to analyze it is methods of analysis. Here we cannot use simple UV methods for most of the herbal drugs in formulation form or in extract or fraction form. So what we have to do? We have to go for HPLC or LCMS analysis of that particular formulations. And first we have to develop the methods for that formulation, and then we can use these HPLC methods or other LCMS methods for these formulations. So this was the study which we have carried out by using UV analysis. Even though we got that good R-square value coefficient 0.997, but still we need to develop that further by HPLC methods. So this is the way how we can separate the compounds from the extract, and we can work out on that particular compounds by checking its some properties by instrumental methods of analysis. This is the different combinations that we have used for this, and that better formula that we got from that, and we got a good particle size over here. Already we have done TM images for this purpose. Actually, if you want that surface morphology by using simple SEM, you will get the surface structure of that, and the transmission electron that could help you to identify the incorporated chemicals in your formulation, and depend upon that you can use that one. And this drug release was also monitored, and this stability studies was also carried out for this. For first is for nano emulsions, and later table is given for blank formulations. And here we got the appearance, clear transparent appearance of the formulations, which we got the very less particle size 44 nanometers with -16 zeta potential. And after stability, the particle size got increased 172. and but the interrupted efficiency was remains same. Okay. So this another one. This is another formulation type that we can do for herbals. That is a metal nanoparticle for external use formulation study. Generally, these metal nanoparticles like silver nanoparticles or gold nanoparticles, if you use for anti-infective, antimicrobial study, antimicrobial purpose. Then this is a part of study when we have used green synthesis of silver nanoparticles from lychee plant. I believe that peel of that lychee plant was taken as well as seeds of the lychee plant was taken, which we consider it is a waste material, but it has a lot of potential chemicals present in it, and that could help us to prepare the formulation. This is the external formulation that we have tried to prepare. So here, when we are using this herbal test, already it is well known. It is actually even comparatively old method of nanoparticle formations. A lot of work has been carried out over here. So that here we have tried. Already it is known that the chemicals like phenolic compounds present in the plant that could have some capping agents in case of metal nanoparticles, and that form these metal nanoparticles well. And already chemical methods are also available by using reducing agents, external reducing agents, capping agents that we can add and prepare the silver nanoparticles. Difference in this study is that we are using plant-based chemicals. Those plants we can use that are edible, which we can eat or which could be the food. So if you use that, that could be safer than the externally added formulations over here. Okay. So here you see lychee peel as well as lychee seed extract we have used, and that was studied for its silver nanoparticle formation naturally with the sunlight as well as heating. These are the two normal, basically two methods that we can use. Simply just we have to put this silver nitrate solutions along with this drug, that we will get this nanoparticle formation in sunlight. Okay, within 5 minutes, just we start to prepare this nanoparticle. The concentrations of the nanoparticle will be varies. If you keep it for longer period of time, then the concentration of the nanoparticle will increases. So further, we have also studied that actually we have removed reducing agents from that. We have removed phenolic compound from the extract, and then we have tried to prepare the nanoparticle that was not prepared, and that could conclude that only the reducing agent that we already determined, we know present in the plant, that is helpful for preparation of the silver nanoparticles. So the method used was sunlight and heating method. These are the features of these two nano formulations that I have prepared. One is a peel and one is a seed, simply by sunlight method as well as heating methods. So this is a heating method. This is the lychee peel LP is given there, and the lychee seeds extracts are there. Okay, so initial appearance of the extract was like that. When you allow it for heating at 70 degrees Celsius, then it turns into this color, and the preparation of the silver nanoparticles can be analyzed by if your solution get brown color. Okay? And already the silver nitrate is the colorless, you know that, and it turns this color of the solutions into brown color. So you can immediately say that there is a formation of the silver nanoparticles. Further, you have to analyze it, silver nanoparticles. So there is UV methods, given methods is there for preparation of the silver analysis of silver nanoparticles. So the first A graph that you see, this is for UV analysis absorption between 200 to 800 nanometers. which does not show any peak at around 420 or 400 nanometers. So that indicates this is a range where we conclude the synthesis of silver nanoparticle. So in B, there is a formulation with a lychee seed as well as C with a lychee peel, that we got this peak due to the formation of silver nanoparticles. And we have monitored this in sunlight as well as in heating at different time interval. So you see in below peaks, UV peaks, if you increase the time period, then the nanoparticle formation that increases. The peak that indicates the concentration of height of the peak indicates the concentration of the nanoparticle that you got. But at certain time, it gets stopped. Okay, so you have to, if you try to identify that certain time that there is no more higher rate of formation of nanoparticles, then you can stop that synthesis of nanoparticles. Okay, this was the study of nanoparticle formation, particle size of the silver nanoparticles. Actually, we have recovered that silver nanoparticle. One more step, important step over there is any nanoparticle, if you prepare, that should recover from solution form. Okay, it is there in solution form. Mostly, if you are preparing this solution or liquid nano formulations. So from that solution, you have to recover it just by one or two ways. Simply, you have to remove any unwanted substance than the nanoparticles. Okay, there are a lot of aggregations can happen. Aggregations can form in your formulation. That could be from excipients, or that could be from interaction of drug and excipient, or that could be from solubility of drug into your formulation. Okay, normally in most of the formulation, what we use the different solvent evaporation method, where we are dissolving, solubilizing our drug into one solvent, and we are adding it into the other solvents, which is a opposite polarity. Means hydrophobic materials we are adding into the hydrophilic or water-based materials, which are stabilized with PVA, or which are just added with surfactant, so that the nano formulation will form. But in this case, what happened? There could change in the solubility of the drug material. Okay? So if it get insoluble, it form the precipitate, it form the aggregates or precipitate. It will remain in the precipitate. So we have to remove the precipitate. Otherwise, it will interfere into the entrapment efficiency, determination of entrapment of that drug. So initially, you can use centrifugation technique to separate it. For example, around 1,500 to 2,500, you can centrifuge it for 15 to 20 minutes, so that you will get settled particles of higher density. The particles get settled. That particles are of mostly for from that aggregates, okay, which insoluble materials remains into the solution. So you can take your upper solutions which has a nanoparticles, and you can separate this lower part, which is aggregates. We don't consider as a nanoparticles in solution form. Specifically, I am talking about. And further, you can separate this nanoparticle by again centrifuging it for higher speed, around more than 20,000, 25,000, 30,000, around 25 to 30,000 RPM. Here you require that particular instrument to do this type of process. Ultra centrifuge instrument you have to use for this purpose, so that it will get around 25,000 RPM, 20 to 30,000 RPM. And you do it for 20-30 minutes or half an hour, then you will get the separated nanoparticles form, and you can remove the upper solutions which does not contain any nanoparticles, and you can rinse it. You can wash it with either buffer solutions or water solutions as per your requirement, and then you can redisperse it into the buffer solution or water solutions, and then you can measure the particle size of that formulation. So this is the particle size which we have measured by this way, and this lychee we got the particle size around 190 from sunlight as well as around 120 from heating method. And that's for seed is 99.40 and around 79 from heating. So we could easily understand the formation of these nanoparticles. So these are the same and same images of these nanoparticles that we have produced. Right hand side you can see there's blank, which is for blank formulation. Background pics are there. Sorry, background image are there in SEM. Sorry, in TM, and in SEM surface images, here are the particles, that how it look like for lychee seed hitting metal nanoparticles and lychee bean by hitting metal nanoparticles. So below are the images for HRTM. Already, if you are interested to know whether it is hardcore form inside the nanoparticle, or it is some core exist and drug which are present over there that exist. That for this purpose, to analyze that, you can use HRTM. Actually, this is a silver nanoparticle. It is actually hard core is there. No any core or space observed inside the nanoparticles. Okay, so this indicates that there is a formation of silver nanoparticles. The another method you can use for formation of this. Okay, this is the figures for TM image for these heating methods and the TM images for others. So these are the very fine images taken at high resolution to check general particle size of it. Actually, in case of zeta sizer we use that use average particle size. Actually, I have quoted that figures above, that average particle size of the solution that based on LSD scattering laser scattering principle. And here individual particle is count. Actually, to determine the average particle size, we have to count more and more particles, and that was not done over here. It's not normally possible to count for a longer period of time, because the instrument is very simple. We can do it, but it takes a longer time to do that. So this is another experiment we can do to check whether there is a formation of this appearance of the silver. If in initial stage, if we remove the solution from the silver nanoparticles, and if you take only nanoparticles from it, then there is no chance of getting any silver nitrate present over there. So we have to remove that silver nitrate by washing at the level of let's say centrifugation. But and then if you take this sample over here, then we could understand that there is a formation of the silver nanoparticles. So the contribution of the silver over there can be determined easily. So this was the study that we have used this type of experiment, that we have removed, we have detected the phenolic compound, that is gallic acid equivalents as well as quercetin equivalent chemicals that we have detected by well-known methods, and we have removed that one, and again we have tried to prepare the formulation, but that couldn't form. So this is a synthesis of nanoparticles. This figure that indicates after removal of phenolics and synthesis of nanoparticles, that is before removal of phenolic, that clearly indicate there is no formation of silver nanoparticles when you remove that phenolic compounds from the plant. So for this we have just used this one. We have used this for antibacterial activity. So we got this antibacterial activity of that, and that conclude that secondary metabolites which are present over there, which are phenolic, that is responsible for formation of the silver nanoparticles, which acts as a capping agents. And the method that already we have used is the green approach method that I have to follow. Okay, this was our research group that carried out this work. Okay, so this is a little bit information about that applications of nanoparticles in synthesis, development of drug from herbals. Similarly, as your allopathic drugs, we can use the herbals for synthesis of nanoparticles, and that could have better efficacy in case of cancer or infectious disease. We could have some success if you go to develop further formulation and eliminate, you can say, toxicity, toxicity of that formulation. So thank you. So these are the contribution of study from our department. So thank you very much. If you have any question, then student, you can ask, get out. Yes.
Hello, sir. Sir, what are the challenges of developing nanotechnology in medicine?
Challenges like this I have explained here. Bioavailability, solubilities are they are basic challenges, but most challenge is the stability of the drugs. When you are using allopathic drugs or synthetic drugs, or there are more challenges in case of herbal drugs than the allopathic ones. The most challenge of herbal drugs preparation of the nano formulations is that the method that we are using for analysis is somewhat tedious, and we have to work on more and more in that case. And normally we use either extract or fractions, or even we have used that reach fractions, that particular chemical reach fraction. Then still there is a problem of analysis in case of intracontact efficiency, or in case of you say dissolution study, or for kinetics of that particular drug in vitro.
Thank you, sir.
Yeah. Yeah, any other question? Yeah, please.
So, what are the future advancements of nanotechnology?
If you are asking about nanotechnology advances, we can here you see we can control this drug release by some other factors like magnetic control drug release, or we could have the target specific drug release, or we could have that drug release at the inside the cell, inside the nucleus, where just I have given reference that if you want to control the cancerous cell development, so we need to work on more and more on that cascade cancer formations, where we can add the false agents, false chemicals similar to that DNA, RNA sequence, and we can work on that. And for this purpose, that material that we used to carry out drug is very important. Apart from that, targeted drug delivery by using responsive materials are also important over here, which we can work more.
Yeah, thank you, sir.
Sir,
yes.
How is nanomedicine looked by regulatory agencies?
Yes, it's a very it's not normally accepted by regulatory agencies. We have to work on that because of the stability issues of material that nanomedicines. So if we try, like phytosomes are one of the case where it is accepted, and it is patented technology, phytosomes, where phytochemicals are added into that. Similar liposomes are also tried, which are accepted by the regulatory, but there are still problems for a lot of chemical, a lot of nanomedicines to accept by the regulators.
Okay, sir. Thank you, sir.
Yeah, any other queries or anything you want to elaborate? Yeah. Anything else you want more details?
Okay, I think there is no queries.
Yeah.
So we could proceed to the further session. Thank you, sir, for elaborating on the topic. Your guidance and experience will definitely boost our creativity and evolve our skill sets. Also, it will create inquisitive thinking and reshape our career. As we reach the end of today's conference, we hope that the newer inventions, innovations, technological aspects in the upcoming years will create new opportunities, enhance healthcare sector, create employment, and lead to a better global health. Thank you once again. I now request everyone to fill the feedback form of the session by the link given in the Q&A tab. So further, I invite Dr. Jayashree Mahore ma'am to propose the vote of thanks for today's international conference.
Good afternoon. I, Dr. Jayashree Mahore, Associate Professor, Department of Pharmaceutics, extend my heartfelt gratitude for making this international conference a resounding success. Shaping the Future of Health, an international research conference organized by SWALife in collaboration with Center for Drug Discovery and Development, Sinhagad College of Pharmacy, Vadgaon, Pune. It has been an enlightening and enriching experience for all of us. Visionary leadership and commitment of Honorable Professor M.N. Navle, Founder President, Sinhagad Technical Education Society, Pune, Dr. Sunanda Navle, Founder Secretary, Mr. Rohit Navle, Vice President HR, Mrs. Rachna Ashtekar Navle, Vice President Admin, Sinhagad Technical Education Society, Pune, have paved the way for remarkable advancement in academic and administrative endeavors. Your support has empowered us to take bold steps, implement innovative ideas, and create an environment that fosters creativity, learning, and personal growth. I would also like to extend my gratitude to Dr. Rajesh Patil, Principal, Sinhagad College of Pharmacy, Vadgaon. Your belief in importance of academic and scientific exchange has made a significant impact on the success of this conference. First and foremost, I express my sincere appreciation to all the speakers for sparing their valuable time. Dr. Lakshmi Balasubramaniam, Consultant Bioimage Analysis, CCAM Bangalore. Dr. Pramod Kashid, Senior Director, Clinical Trials Management, Midspace Singapore. Dr. Kishori Apte, Founder, App Research Foundation, Pune. Dr. Andrea Otto, Founder, OACPI Limited, Italy. And Dr. Deepak Raut, Professor, Pharmacognosy, Amrut Vahini College of Pharmacy, Sangamner, who shared their knowledge, insights, and research findings during this conference. Your expertise and dedication has immensely contributed to exchange of ideas and advancement of knowledge in respective field. Your presentations have been thought-provoking and inspiring as well. Thank you. Thank you one and all. I would also like to extend my gratitude Dr. Praveen Bade, Founder, SWALife Biotech, and Associate Professor, Department of Pharmacology, SUP, and Mrs. Ashwini Bade, Founder, SWALife Biotech, for their instrumental contribution making this event possible. I'm thankful to Ms. Nishigandha Jadhav, Ms. Nehal Veer, Ms. Saili Murkar, Ms. Riya Khede, and Mr. Sujit Chauhan, Junior Researchers, SWALife Biotech. My faculty colleagues from Sinhagad College of Pharmacy and volunteers who work tirelessly behind the scene to ensure the smooth execution of every aspect of this conference. Your dedication and hard work have been commendable, and I'm grateful for your unwavering commitment to the excellence. I would like to extend my gratitude to the delegates and the participants. Last but not the least, I extend my thanks to all the attendees who actively participated in the discussion, asked insightful questions, and engaged in lively debates. Your active involvement has made this conference truly interactive. So as we conclude this international conference, let us carry forward the spirit of collaborations, knowledge sharing that we have witnessed here. The connections we have made, the ideas we have exchanged, and the insights we have gained will undoubtedly lead to further progress in our field. So once again, thank you one and all for the part of this event. We look forward to meeting you all again in near future. Let's continue to work together in the advancement of science. Thank you. Thank you one and all.
Thank you, ma'am. The certificate of participation will be generated by filling the feedback form that is given by the link in the Q&A tab.
Excuse me, ma'am, where is that feedback link? I am unable to see that link.
It's in the question and answer tab.
It is not visible, sir. Question answer tab is not visible for us.
Okay, it's not visible.
Yes, sir.
Yes, sir, it is very much there. It is there.
It is there.
Where, sir?
The question and answer tab, if you see at the top side, in
Where,
the
sir?
question and answer tab, if you go in the question and answer tab, you will find that. Okay. Before finishing, I just want to give a short vote of thanks from my side as well. Just I want to thank two more people from the team. The Coology department of Siagarh College of Pharmacy, the students who have actually played important role. Also, I like to thank the organizing committee, especially Swarda Madam, Shinde Madam, Aishwarya Madam, Sonali Berde Madam, Rutuja Mule Madam, and Pranali Madam, who has actually played important role, and also our non-teaching and teaching team from SCOP. Definitely thanks to all the audience who has actively participated. Thanks. I would like to now request everyone to switch on your cameras. So we'll have a group photo. Thank you all. Now I'd like to conclude the sessions. Thank you.
Sir,
Sorry,
if
I
you
have
could
this.
please send the link for the feedback in the mail, it will be beneficial because even I am not able to see it in the question and answer session.
Okay, yeah, sure. I will email it.
Yeah, thank you so much.
Thank you.
संपलं ना?
Where the feedback link?